Okay, I think we'll get started. I want to thank you all for coming out on this rainy day for our monthly seminar series. And before I get into the introduction of Dr. von Rechem, I have a few announcements. First off, I want to remind you of the seminars we have planned for the next couple of months. Next month on November 7th, we have Dr. Frank Marini coming from MD Anderson. He's going to be speaking on mesenchymal stem cells in cancer, and we'll be addressing stromal effects and also gene delivery attributes of MSCs in cancer. December 12th, we're going to have a seminar by Dr. Kumar Alagraman, who's a faculty member here at CASE. He's going to end the year for us by telling us about a new type of stem cell that he and colleagues have been responsible for identifying from the cochlea. So you'll all want to come and hear about that. In January 9th, we're going to be having a presentation by Dr. Stan Gerson, the director of the center. He's going to give us a sort of a year in review and projections on what we're planning for 2008. And in general, what we've done for our January seminar presentation is also have a poster presentation. So any research posters that you've presented on stem cell work this year, second half of the year, that you would like to share with your colleagues here, please plan to bring, and we'll have our reception and a poster session. A couple of other items. We've just issued a request for proposals for our pilot projects program. The proposals are due on November 26th. We will have a group of researchers from the center do a review of the proposals, and there's a review sheet if you want to see how what factors and characteristics are going to be weighted. And we intend to announce the 4 awards. They're $50,000 for 1 year for pilot as of December 18th. We'll have January for getting those accounts set up with the projects being able to be funded as of February 1st. Also, I'd like to bring to your attention that we have an upcoming opportunity. The Third Frontier program is anticipated to issue requests for proposals for new BRCPs. The RFP should come out a little later this month. The proposals will be due in January. They're going to be smaller awards than what we've seen in terms of funding in the past. They'll be $2.5 to $5 million. So, and they have to be non-overlapping with what we've had support for thus far. So if you have some ideas about the kinds of programs we could put together, perhaps in targeted therapeutic application areas, please think about those, talk with Stan or myself or Paul DiCorletto, and we'll see what we can do to start trying to see what kind of groups can be put together. Please note these really do have to have a demonstrable tie to clinical and commercializable endpoints. So keep that in mind. Also today, for the first time, we're trying a video feed between here at CASE and over at the Cleveland Clinic. We're going to see how that works. Regardless, we're going to at least be able to archive this presentation, and it'll be viewable online on the NCRM website, hopefully by next week. So those are my announcements. Now, let me get on to introduction of our speaker today. Dr. Horst von Rechem is an assistant professor here at Case Western Reserve University. He's in the Department of Biomedical Engineering. Horst started off his academic career at Rice University, where he got his undergraduate and master's degrees in chemical engineering. Then he moved to University of Utah, where he pursued his doctoral training in biomedical engineering. and then decided to see how things were on the East Coast. So he did one postdoc at the MIT Harvard Medical School in Boston. Guess he decided the East Coast, wasn't quite sure if he liked that coast, so he moved to the West Coast. Did a second postdoc at University of Washington in Seattle before settling here at the North Coast as an assistant professor in 2005. Dr. von Rechem has been very prolific in the areas of biomedical engineering and interfacing between biology and scaffolding. He also has experience in the use of embryonic stem cells, and that's what he's going to tell us about today.
So I also want to thank you all for braving the rain. As Deborah mentioned, I came from Seattle. This will be nothing in Seattle, but I understand by Ohio standards, this is quite a endeavor to come through. So I was going to talk today about embryonic stem cells. And Deborah had also wanted me to talk a little bit about a workshop I went to in Stanford where we discussed escrows. Escrows are embryonic stem cell research oversight committees. I'll spend maybe a couple minutes at the beginning sort of discussing, well, what exactly are embryonic stem cells in relation to the adult stem cells, which most of the CSCRM work centers around. And then I'll talk a little bit about the escrows. I have a slide that says discussion. If you feel we need to discuss at that point, maybe we can. Otherwise, maybe we can hold the discussion to the end. But the bulk of my talk, I think, will be about specifically about my research, as Deborah mentioned. So stem cells, most of the American public sees stem cells as this one big category, and maybe distinctions aren't quite clear. CSCRM works with adult stem cells, these multipotent stem cells, things like mesenchymal stem cells, hematopoietic stem cells, and you're all very familiar with these. That we can derive a number of different tissues from these kinds of cells. One sort of issue, particularly for people who work in the embryonic stem cell field, is we kind of feel that maybe not every tissue is capable of being derived from currently known adult stem cells. There's a lot of great progress in looking at new stem cells. I'm actually interested in this cochlear stem cell that Deborah was mentioning. But you can see, mesenchymal stem cells have their potential, but there are all of these adult stem cells. Oh, thank you. All of these adult stem cells are what I'd say somewhat, I may get in trouble with my colleagues, I say somewhat limited applications only because we don't necessarily know what is a stem cell to create every given cell type. So I've divided this box that was a stem cell box into two boxes. So the adult stem cells, ones that you're all very familiar with, and the embryonic stem cells. And the embryonic stem cells, because theoretically we can derive every cell line from an embryonic stem cell, I could say that they're potentially unlimited in therapeutic applications. But in reality, we all know that there's going to be many complications, including ethical complications that we'll discuss a little bit with these research oversight committees, and other biological complications that make it difficult to get exactly the cell that you want, which I'll discuss in my research talk. So one thing I want to point out before we go too much further is if you're not too familiar with embryonic stem cell research, even this one box can be subdivided into two boxes. So many of you may know that President Bush in 2001 created some legislation on what kind of research the federal government will support. And there are approximately 21 what are called embryonic stem cell lines that were considered at that time acceptable, and from then on, were deemed usable and able to apply for federal government funding for research support. These are human embryonic stem cell lines. I work with one of these human embryonic stem cell lines. 21 is kind of a nebulous number, I think, when they actually got down to trying to figure out how many of them were alive and working fine. They're, I think, down in the teens now. But the issue was that there were certain lines that were created prior to 2001 that President Bush said, okay, we made these, we might as well learn what we can from them. Then there's sort of a gray box, which is all other work with human embryonic stem cells. But the important thing to distinguish is that this, in the US, this is research which is not federally supported. So, well, what kind of research fits into that box? One type of research is creation of new lines. Particularly, I'm sure you've heard of this initiative in the state of California where Arnold Schwarzenegger is paying out of his own pocket to fund, I think this year, $300 million worth of creation of new embryonic stem cell lines. And the idea is that maybe not everything was capable of being discovered in these original 21. You know, there might be a lot to learn in creation of new embryonic stem cell lines. The other category that I break this down into is embryonic stem cells from a patient's own cell. So this is some kind of therapeutic cloning. Not necessarily a lot of research going on in this country, a lot of research in Japan, a lot of research in the United Kingdom, and we had also probably all heard of the research that had been going on in Korea and sort of the fallout that came out of that. But I just wanted to demonstrate that there's a number of different areas that we can work in embryonic stem cells with different levels of sort of funding and public interest and maybe ethical considerations. And in light of that, as I mentioned, I went to a workshop in Stanford on the formation of what are called embryonic stem cell research oversight committees. These are escrows. And the idea was that in about 2005, the National Academy of Science created these guidelines that said any school, any university, any agency that's conducting embryonic stem cell research should voluntarily create some kind of organization to provide oversight for the research going on in that field. And these would be similar to things like the IRBs, which provide oversight for any clinical research, or IACUCs, which provide oversight for any animal-based research, animal oversight committees. And this kind of oversight would be specific to proposals involving embryonic stem cells. That's the escrow part of the escrow. And so their guidelines were published, I think at least the outline is freely available from this website. So if you're interested in seeing what the National Academy of Science had to say about it, their idea was that if we voluntarily start oversight now, that this could circumvent or bypass the need for maybe a more restrictive government agency to mandate oversight on us later. Similar recommendations were published by the International Society for Stem Cell Research, ISSCR, in Science in 2007. And the important question that we addressed in this workshop was, which research does need oversight? So, this field I already showed you has a lot of diversity. So what could we, should we, do we need to oversee? And the question is, should it be all stem cell research, that big box, not just the embryonic stem cell research? And in California, actually, they had decided that all stem cell research of any kind, adult stem cells, embryonic stem cells, should be addressed by an oversight committee. And they're actually called SCROs and not escrows because they're stem cell research oversight committees. Based on these guidelines, based on these NAS guidelines, it's possible that we might restrict our oversight committees to just embryonic stem cells. And then there may be issues that aren't necessarily dealing with creation of new embryonic stem cell lines or use of embryonic stem cell lines, but maybe traditional research that's going on in our universities all the time, like creation of transgenic animals uses, for example, mouse embryonic stem cells, or examination of a stem cell potential using teratomas. We may want to be asking ourselves, should our escrow be evaluating only the use of human embryonic stem cells? And so this may remove the question of whether animal stem cell research should have this level of oversight. And if so, do we include all human embryonic stem cells? Do we include these federally approved lines? Do we include only discussion of new creation of new lines? What about some of these new areas that are currently sort of being investigated or coming to the forefront of the public attention? Things like these induced pluripotent stem cells, where researchers were able to introduce genes and take adult mature cells and bring them back to a more primitive embryonic state. Should that require this level of oversight? What about chimeras, where cells from one zygote are transplanted to another zygote? So chimeras, unfortunately, received a lot of negative press recently. There was, I don't remember where I saw the cover. I actually tried to Google it before I came. There was a journal cover that had a sheep with a human face on it, and that was probably intended to create some negative press. But certainly, I think when we think of chimeras, that's probably one of the last things that comes to our mind. But a chimera, by definition, is any individual that has cells derived from more than one zygote, so more than one diploid organism. And we see these all the time, so these aren't necessarily a new thing. Bone marrow transplantation has been going on. I came from Seattle, so that's where a lot of this was pioneered for decades now. Mouse transplantation for examination of human stem cells and progenitors even goes on here, many different departments. Use of human immune cells to model the immune system in SCID mice. These are by definition chimeras. Should these be involved with these research oversight committees? Examination of teratomas, the developmental potential of of stem cell lines, be it human, be it other species. So I kind of listed this in a level of maybe complexity, maybe challenges in sort of questions in ethics. There's work even on looking at differentiating stem cells and recreating things like the central nervous system in higher order animals. This is a more complex chimera than we're probably used to, but is this the level that we need to address our research oversight committees? And so another question is, what kind of guidance do we get? Fortunately, we have a lot of good guidance here already available. Our former president, Greg Eastwood, is now the director of this Inamori Center for Bioethics and has been very interested and involved in discussion with me about this concept of these escrows. Dr. Eastwood and Insoo Hyun is a bioethics professor here at Case who's been very heavily involved. He was very fortunate to be able to visit the lab in Korea where the human embryonic stem cell creation transplantation was taking place and has a very unique perspective on what was going on, and has been involved in a lot of publications in the area. These are some publications that Dr. Hyun sent me on some of these more controversial areas like creation of chimeras, these induced pluripotent cells. And both Dr. Hyun and Eastwood have been very interested and involved in discussion about creation of these embryonic stem cell research oversight committees. And so that's sort of my feedback from what went on and sort of what we discussed. And I'm open for discussion. I don't know if anyone feels urgently needing to discuss now, or if we want to just segue right into to some of my research talk, and then we can sort of address both kinds of questions at the end. I don't see any hands, so I'll move forward. So my research specifically, working with these stem cells, is examining taking tissue-specific populations out of differentiating embryonic stem cells. So what do I mean by that? Well, many of you might be familiar with adult stem cells. So this is a typical diagram for stem cells. The definition of stemness means two things. Typically, one is the capacity to self-proliferate. The other is the capacity to form more mature tissue. So here I have a stem cell, and I can form one tissue type. I can form other tissue types. With adult stem cells, the big concern is typically a question of proliferation, right? How do I get enough cells from a very limited source? So we don't have an unlimited source of stem cells in our body. How can I get enough cells out of that? Sometimes even questions of how can I get them to proliferate once I get them out? And in that sense, how do I maintain this pluripotency? If I take them out and I cause them to proliferate, will I lose some of this capacity? And so one issue with these adult stem cells is, this maybe limit. I cross it out. It's not no proliferation, but maybe more limited compared to embryonic stem cells. But the good thing is that oftentimes, I would say relatively speaking, directing one tissue type is oftentimes fairly easy. So I think a number of different researchers here at Case have demonstrated that adult stem cells can be directed to a specific tissue type for a very usable therapeutic application. Embryonic stem cells, unfortunately, have a very different problem. Proliferation is easy, very fast. We do clonal expansion where a single cell can form 10 to the 9th cells in a very short period of time. So proliferation is not really our problem. Our problem is control of differentiation. So we can proliferate, but it might be hard to direct it into one tissue type. The tissue type we're interested in are endothelial cells, and our concern is, how do I create a population of only endothelial cells and not undifferentiated cells or not cells of another tissue type? And so the problem with embryonic stem cells is when I remove the factor that prevents differentiation, I spontaneously form all three of these different populations. And our challenge is, how can I select this one population that I'm most interested in? There's a couple ways people have been looking at this, certainly not an exhaustive list, but maybe the more popular ways. And I might sound a little sarcastic here, but the sort of take-home message, if you take nothing else home, is it's extremely difficult, right? So what I say is, how can you find the right mix of differentiation factors that will result in 100% differentiation? So can you take all of these and make only those? And if so, how can you do that? What's the right mix of soluble, insoluble things? Difficult thing to do. Another possibility is, what if I could find just the right antibody that maybe could allow me to select and pick out only the ones I want and leave the ones I don't want behind, picking out through things like FACS or MACS. We'll talk a little bit about FACS in this talk. Another area that I was investigating when I was at the University of Washington is, what if I could create a suicide switch that would kill these undesired cells. And so my cells that I'm interested in would survive. An area that I'm interested in here at CASE that I'm working on is what about finding a way to get proliferation advantage, so the cells that I'm interested in can outcompete the other cells that I'm less interested in. And so to do that, we're doing some genetic modification of these embryonic stem cells. So that takes advantage of the fact that the embryonic stem cells have this very high proliferation capacity. I can introduce genes, and I can patiently, over time, select cells, clonally select cells that have the genes that I want working how I want them. And so the genetic modification that we introduce has two important components. One is some kind of drug selection that's under constitutive expression. It's always on. And so I can select which cells got the gene, the DNA, the information that I'm interested in from the population that maybe didn't get the gene. The second piece of genetic information that we're introducing is a marker, some kind of identifiable signal that's under what's called a tissue-specific promoter. Promoters are these regulatory elements that turn on in specific tissue types. And so if I can use something like a green fluorescent protein under control of a promoter such that it only turns on when a cell turns into endothelial cells and doesn't turn on into other tissue types, now I have a self-identifying cell. And I can find a way to selectively pick out that cell or do something with that cell and allow it to be removed from its undifferentiated or differently differentiated neighbors. The specific promoters we're using, this is the only time I define them a little bit better, so if you're not familiar with them, I apologize. FLIC is a receptor for the vascular endothelial growth factor. TIE is a receptor for another mitogen, angiopoietin. PCAM is the PCAM promoter is for the platelet endothelial cell adhesion molecule, and VE cadherin is another cell vascular specific adhesion marker. And so we've taken the promoters, I want to make sure that's clear, it's the promoters we're using, not the genes, the promoters, and allowing those promoters to control the expression of this selection gene, in this case GFP, green fluorescent protein. So here I'm showing, you'll see a few different constructs. So here's the promoter for FLIC controlling the GFP gene, and here's this other piece of information, this constitutive selection marker that allows me to select whether the cells got this DNA. Here's PCAM. You can see the range of sizes of these promoters. TIE is the angiopoietin receptor. Again, the GFP controlled by these promoters, and then this constitutive selection to allow me to select for cells that got the DNA. So once we've created these constructs, the first thing we wanted to do was to test, are these constructs actually working in cells that we would expect them to work in? So we tested a few different cells. We tested human umbilical vein endothelial cells, bovine aortic endothelial cells. The idea was take an endothelial cell, we know it should have these promoters on, introduce these genes, and sure enough, they came out green just as they're supposed to. So here's a few, that was the FLIC, I believe. Lost some text due to formatting, but here's PCAM, here's TIE. So all of these resulted in green positive endothelial cells under control of these promoters. The other important question was, are they going to be off when they're in negative cells? So cells that are not endothelial, are they shouldn't have these promoters on, are they therefore not green? And it's kind of boring to show you a picture of cells that aren't green, so one thing we did was compare FACS expression profile of 3T3 cells. These are a fibroblast immortalized cell line that did not receive any DNA with ones that were positive, stably positive for this TIE promoter GFP construct. And you can see there's little to no difference in background GFP expression on these non-endothelial cells. Okay, so we've tested on these cell lines, these immortalized cell lines or endothelial cell lines, but I was mentioning that we wanted to really look at embryonic stem cells. So what embryonic stem cell line do we use? Well, we're doing, as I mentioned at the beginning, we're doing a little work with human embryonic stem cell lines, one of these federally approved lines. The issue with these human cells is they are about 3 times slower in proliferation than mouse cell lines. And so when you talk to a new graduate student, you say, okay, do you want to work on your PhD for 5 years or do you want to work on it for 15 years? They usually opt for the 5-year plan. And so the 5-year plan in this case includes the faster growing cells, which you're allowed to laugh, that's okay. which was a mouse embryonic stem cell line. In this case, it was a stem cell line called E14 TG2A. We'll usually call it E14. And the nice thing about these was that they maintain the full differentiation potential. They're one of the first cells to demonstrate this without the need for feeder cells. So feeder cells are these cells that in many cell lines, many embryonic stem cell lines require to support and maintain undifferentiated state. These E14 cells don't necessarily need those. So we introduced the DNA by electroporation. Somewhat low efficiency method, but the nice thing is we have unlimited cell proliferation, so we can take time and clonally select cells that have stably integrated the DNA. And then through a couple of different differentiation protocols, these aren't necessarily 100% pure differentiation. These are, as good as we can do. We tried to direct differentiation into embryonic stem cells. The protocols often involved administration of an endothelial-specific mitogen, like VEGF, and often involved a certain wait in time. So endothelial cells, typically in vitro differentiation, are seen around between day 5 and day 7, depending on the marker you're looking for. And they're maintained in this medium called EGM2, which is one of these sort of magic mixes that people created to try to optimize endothelial differentiation. And so what do these E14s look like? Well, here's a couple of different images showing some of these constructs. And what you see are cells, embryonic stem cells, that have differentiated. Again, I mentioned they go into all directions. Some of them become endothelial cells. Some of them become other cells. And you can see that these islands of cells that have become endothelial cells are GFP positive, whereas the background cells that look maybe more like a fibroblast lineage are negative. Similarly here, even within a region of cells, some of them will be positive, some will be negative. This is using the PCAM construct. Similarly, we saw with the FLIC construct and the TIE construct that you could selectively get populations of cells that were GFP positive. Presumably, this is still presumably due to endothelial differentiation. So we want to do a little bit more analysis on these cells. So one very high throughput way we can analyze these cells is through fluorescent activated cell scanning analysis, where we can have the cells come through a detector one at a time and evaluate their fluorescent properties. And I mentioned earlier that many people are interested in using FACS to pick out populations of differentiated cells. Well, what we've already created at this point is self-identifying cells. So I don't need to add any antibodies. I don't need to add any reagents to identify which cells are endothelial cells. Hopefully, these endothelial cells are already identifying themselves because they're green and fluorescent and can be picked up by FACS. And that's exactly what we saw. So the bovine aortic endothelial cells, these are our controls. Without any DNA, this is a FACS profile. This is the green fluorescent channel. We see that once we introduce this gene, this GFP driven by the TIE promoter, we have a large population that's GFP positive. When we introduce PCAM, that population gets even larger. For embryonic stem cells that have stably introduced these different constructs, here is the TIE construct, the FLIC construct, the PCAM construct. At day 0, we don't expect any or few endothelial cells. We expect most of them to be undifferentiated. So then that's the case. That's what we see. By day 4, that's early. That's right before we expect to see any endothelial cells. We might see a few, but depending on the construct we use, by day 8, 6, or day 7, we start to see a population of green fluorescent cells sort of self-identifying in this mix of differentiating cells. Problem is it's extremely messy. So there's two issues. One is that typically we do have a background of starting cells where some cells are already differentiated, and so we can't ever start with a pure population of undifferentiated cells. Then the time course of differentiation varies. So some may differentiate immediately, whereas some may remain undifferentiated and only differentiate later. So we see things like big error bars and a very messy process. But we can detect some trends, things like PCAM, which is a late endothelial marker showing up much later than FLIC, which is an earlier endothelial marker. TIE, which is also a sort of a middle endothelial marker, we can see showing up at about day 5, day 6, which is what other investigators have shown to be about the time that TIE expression, the TIE gene expression, is activated. What I want to show with this is how incredibly complicated it gets depending on how much pre-differentiated cells I have in the mix. And initially, we'll always see an increase in the number of endothelial cells, but over a period of time, we'll always also see a decrease in the number of endothelials, in the relative number of endothelial cells. So here you can see these are within this graph. I have another graph, I don't know if I included it, where we carried these out to about 30 days. They continue to go up and eventually go down as well. And what we believe that is going on here is that although the cells are self-identifying, they're not able to outcompete the undifferentiated cells. So here we have a differentiation signal directing endothelial differentiation, so we have an increase in the endothelial population. And over time, the undifferentiated cells, or the cells that have differentiated into another phenotype, continue to proliferate and maintain their proliferation status and are able to outcompete the endothelial cells, which have a reduced proliferation capacity. What we said is, well, how about if now that we have these cells that can, actually I noticed that we have a student here from Zhenghong Li's lab who's interested in modeling this for us. You know, what happens with the undifferentiated population, what happens with a differentiated population. We think it can kind of be an interesting model to say the time profile of differentiation and the activation of these different signals. Sorry, side note, plug. We have these cells that self-identify. What if we were to take a fax, but this time a fax sorter, a cytometer, and pick out the GFP positive cells? So we did that. We sorted for GFP positive cells under these various conditions, picked them out, and from heterogeneous populations, and then examined them with a number of different things, such as their capacity to uptake low-density lipoprotein. This is not necessarily a unique endothelial property, but endothelial cells have this capacity. Some hematopoietic precursors also have this capacity to uptake these low-density lipoproteins. The nice thing is it's an easy assay. You just add a reagent and they pick up these particles and turn red. We also examined some antibody labeling, including labeling for PCAM, this peripheral endothelial adhesion marker. Here's the different constructs, the FLIC, the PCAM, I think the last one was a TIE construct, all showing very high low-density lipoprotein uptake. In contrast to undifferentiated cells, which have virtually no low-density lipoprotein uptake. Here's PCAM staining showing just about every cell that's DAPI positive is also PCAM positive. So we've picked these populations by FACS and their endothelial cells. This is showing other constructs. The PCAM construct also has similar results. This is staining for von Willebrand factor, another endothelial-specific marker. These E14 cells are positive for that as well, compared to Huvic cells, our control cells, which show the same level of von Willebrand expression. So the big problem with FACS is that, first of all, FACS is really costly. So if I want to use a cytometer to pick out my cell populations, as a young professor, I can't afford to do a lot of that. The other issue is that FACS is not 100% pure. So I'm an engineer. I tend to maybe critically analyze particularly devices and instrumentation. And I think about a fax and how easy it is. If I slip one cell next to another cell, I could accidentally have a selection of a cell that I don't want. So what we said is, can we find a chemical way or a biological way of self-selection that would remove these fax complications? And so we decided, rather than just use a GFP expression, we decided to introduce a GFP puro fusion. Puro is another drug selection gene. And the idea would be that not only is it GFP positive, but when it becomes an endothelial cell, it is now also resistant to a drug. So after I differentiate the cells, I can add this drug, I can add puromycin, and kill off all the cells that have not become the endothelial cells that I wanted them to become. So we used a similar protocol. We introduced the DNA. I think in my next slide I'll show you the DNA. I did that similar differentiation, and then I added puromycin after endothelial selection at different days, depending on when these promoters turn on. So remember, I mentioned FLIC is an early endothelial promoter, so it turns on around day 4. VE cadherin is a late promoter. And part of the reason we chose these 4 different promoters is we felt that this process would allow us or enable us to pick endothelial precursors or early endothelial cells in a different stage of endothelial differentiation. So we can get some cells that maybe are early or young or primitive endothelial cells, some that might be very mature or more mature endothelial cells, and presumably these would have different functions, different properties, and different capacity to be used for things like tissue engineering, regenerative medicine, which is sort of my main focus. So this is what these constructs look like, very similar, except rather than just the EGFP, we have an EGFP puro fusion. I don't think show any data demonstrating that this EGFP Puro fusion works. You'll have to take my word for it. It was tested in constitutive systems. In bovine aortic endothelial cells, so that's again our control cell, we introduced these constructs with these fusions, and what we see is the GFP expression is a little bit shifted, a little bit dimmer. That's the one limitation of this particular fusion, is that the GFP isn't as bright as GFP by itself. But when I select using Puro, I end up with a population arguably pure endothelial cells. This is our control, so presumably they all started out as endothelial cells. When I use that same system in my E14, in my embryonic stem cells, I end up with a similar expression profile where my day 0 cells, my purple population, should all, not under Puro selection, should all be non-green. Day 3, I don't expect the Thai expression system to have turned on yet, so I still have only non-green cells. And then I don't remember when we added, it was on that other slide with Thai, I think it's day 5, we add puromycin, so we have about 7 days of selection, and we end up with a population that's almost pure endothelial cells, and arguably very few undifferentiated cells or cells of a different lineage, and that persists even day 16. If you remember, day 16 was when proliferation of non-endothelial cells was really taking off. So the last thing we wanted to do was take these cells that we've selected using this Puro fusion construct, and evaluate their identity as to whether they're endothelial cells. And so we looked at a number of different markers. So here are the cells using this Thai construct, examined using FLIC staining, PCAM staining, LDL uptake are these two, I believe, and comparing that to HUVEX. Here's von Willebrand staining, PCAM staining. I have a second von Willebrand, but this is using the Thai construct. Here's a PCAM construct. So this is the PCAM promoter driving the selection gene. Here again, I have FLIC staining, PCAM, von Willebrand staining, and then here's LDL uptake. And LDL uptake of undifferentiated cells, you see, it's still close to 0. VE cadherin construct. I don't have a watch, so I don't know how my time is. It's okay. The VE cadherin construct, also very similar kinds of expression that we have, including our GFP positive. I think this is GFP positive antibody staining. Unfortunately, the GFP is dim enough we can pick it up by fax, but picking it up by microscopy has always been a little tougher with the microscope, particularly the camera we have. Then my fourth construct, FLIC, also showing the same kinds of things. Here's an antibody control showing little to no background staining. So the question is, now that we have these cells, we have this capacity to both self-identify and be able to self-select these populations of cells. Some future directions we'd like to go to, it's one thing to show nice antibody staining, but it's another thing to actually show expression of endothelial-specific function. Molecules that endothelial cells need to be able to do if you're talking about using them for regenerative medicine or therapeutic applications. Things like tissue plasminogen activator, enos nitric oxide synthase, prostaglandins. These are molecules that endothelials naturally secrete biologically to regulate coagulation and platelet behavior. Another thing we're interested in is concurrent selection using multiple endothelial-specific promoters. So is a cell that's both PCAM selected and Thai selected different from maybe the pure Thai population? The FLIC population, for example, to us is kind of exciting because some recent evidence indicates that FLIC is active both in endothelial precursors and in early smooth muscle precursors. And so the possibility is if I'm tissue engineering a blood vessel, I can take a FLIC positive cell and then pick maybe the PCAM positive variety and send that into endothelial, and then pick a smooth muscle cell positive variety and send that into a smooth muscle cell direction. So that's another direction we're heading, involving a ton more cloning and some more diverse markers. So starting to stretch my capacity for molecular biology. We're also looking at, we have a number of collaborations. We're looking at in vivo evaluation of these cells, how they perform. We're working with Zhenghong Li in radiology and David Wilson in my department of biomedical engineering, using some of these self-reported cells for imaging. The idea is, can we see when cells differentiate in the body? There's a lot of interest in self-differentiation as a therapeutic goal, and we now have the capacity to examine by self-reporting as when and where this differentiation occurs. With Keith McRae over here in Department of Hematology, I believe, we're looking at using these cells for angiogenesis. There was a summer project that was kind of a teaser, but very exciting, demonstrating that these embryonic stem cells can participate in tumor angiogenesis. And we could see them and follow them because they had these endothelial-specific markers. The other direction we're taking, so this is a lot of work on endothelial cells. This was for my first student who's been working for me for about 2 years. We have some new work looking at similar strategies in other cell populations that have historically been a little difficult to get from adult stem cells. So one of those are cardiomyocytes, and we're involved in some collaboration with Mark Penn over at the Cleveland Clinic on examining directed differentiation using cardiomyocyte-specific promoters. And motor neuron cells, we're involved in a collaboration with Bob Kirsch, who's in my department, Harry Hoyen, who's a surgeon at Metro, I believe, looking at using motor neuron-specific promoters to do the same kind of self-selection strategy for early motor neuron precursors. And in closing, I'd like to acknowledge the members of my lab who contributed to this project. So Sage did a lot of the endothelial work. Elaine is on the cardiomyocyte project. Thomas Cowan is on the motor neuron project. James is an undergrad who's been helping out. Dr. Margaret Barron, I was fortunate enough to be introduced to when she was interviewing for position here, has been a great help in the FLIC-based constructs. That's one of the sort of areas that she's focusing on. And that's it. So I think I have time for some questions, and thanks for your attention.
Questions for Dr.
We can talk about escrows too if you're. Who wants to serve on an escrow? That's the hard part.
I have a question. How difficult do you think it's going to be to consistently get the ES cells down these alternate differentiation pathways like the cardiomyocyte or the neural?
That's the thing is if you're only relying on some differentiation or lack of differentiation stimulus, the selection is extremely poor. So I think the naturally occurring endothelial cells in a differentiating population can vary in the teens percentage-wise. And so, how do you get a 100% pure population, or how do you even get a 99.9% pure population? Some alternative strategy needs to be made. The directions we're using, there's still problems with it. So no promoter system, I think hopefully a molecular biologist here didn't have a problem with that, is 100% on, 100% off, and so nothing is bulletproof. We're hoping with multiple strategies that we can come up with something that's a little tighter. But the nice thing is that a lot of these constructs that we're using were first used in developmental biology. And so what's interesting is that the constructs tend to be a little tighter or a little more consistent in vivo than in vitro. And so one of the other directions that's sort of on the distant horizon is to design an in vivo selectable strategy so that once the cells are in vivo, they have this proliferation advantage or the survival advantage as compared to the undifferentiated or differently differentiated cells. But I think it's still a work in progress. So I don't think there's a therapy on the horizon tomorrow. But hopefully, maybe some potential at the very least for examining cells for things like tissue-engineered vascular grafts or tissue-engineered vascular beds, where right now it's a little bit difficult to get a high number of cells and evaluate a lot of different strategies. So maybe basic research phase is I think what we're shooting for.
So with the electroporation of your ES cells, how long have you followed the cells until you lose the signal?
So the electroporation, what we're doing is we're electroporating a very large population, somewhere on the order of 10 million cells. And most of them have transient expression. So most of them lose expression about 7 days. And a very small percentage, something on the order of 1 in a million, gets a stable DNA integration. And we've followed these cells out some of them out for almost 2 years. And in my previous lab at the University of Washington, we even did, now I forget the analysis, but we did analysis of chromosomal integration and demonstrated that there is in fact chromosomal integration in a very small population. The nice thing about the embryonic stem cells is because they have this high proliferation capacity, you do have the time and the capacity or the opportunity to be able to select clonally. So if you electroporate 10 million cells and only 10 have integration events, you can wait until those 10 cells grow up to enough cells to be able to use. I'm dinged on every grant proposal when I say that, though, because maybe you're going to head this way, is as repeated proliferation cycles go on, I'm increasing my risk of cumulative chromosomal mutations. So yes, I can go many cycles and do a lot of selection, but the whole concept of mutation and then therefore having a problematic cell is increased as well. So does that answer your question? Yeah.
Okay, you tried the primary cells in like mouse.
So the only primary cells we've tried are these human umbilical vein and bovine aortic. They were primary isolates. The nice thing is they have a good proliferation capacity. Mouse endothelial cells don't have a great proliferation capacity, and it's hard to get them out of the mice. But if that's the area you work in, I'd love to try those cells, because to me those would be a better model. These are all mouse promoters, so they probably would be more suitable in a mouse model. So I'll go back here. Over there.
Yeah, if I get it right, so you did most of your analysis with the pool of the stable transfectant, am I right?
No, with clones. So we would select clones. So So the nice thing is over time, you have this plate of, let's say, 10 million cells. You can follow the individual colonies of single cells that have grown up. You can pick those colonies and separate them and clonally analyze them. And so that's another issue. Particularly the PCAM was the trickiest. Our collaborator who was working in the PCAM area, she also said that only about 1 in 16 has PCAM integrated in a useful enough site that you get enough expression that you can see. So we had to screen a ton of PCAM clones before we got any that were bright enough. So it seems like it was maybe easy and fast data, but it was a lot of clone screening. So yeah, that's a limitation of the system. Please.
So I actually don't know a lot about stem cell research, hence my reason for coming. But
Good.
I kind of feel that there's probably two camps of pro-adult stem cells, pro-embryonic stem cells, specifically certain professors like Dr. Shirley at MIT, who's pro-adult, like very much so. And I know there's some controversy in terms of perhaps embryonic stem cells inducing tumors or some crazy stuff. So what do you feel like? Can you just kind of talk about those issues and when you talk about the future of tissue engineering, do you think that future is 50 years from now?
I'll have a real difficult time evaluating that, because what I thought a student could do in 6 months, that's taking them 2 years, that kind of thing. But I hope the future is closer than we think. I think using embryonic stem cells is probably much further in the future than using adult stem cells. Adult stem cells are used today for many applications. Some of the limitations you suggest, things like the capacity to form tumors, maybe ethical concerns, are probably going to be a big barrier before we see embryonic stem cells used therapeutically. So that's going to be a challenge. The way I see it, so I'm not a clinician, so my primary goal isn't necessarily to get something into clinical practice. I'm more interested in sort of how can we evaluate something, whether something will be suitable for a therapeutic application. And I think that's a big strength that embryonic stem cells can play at this point, is allowing us to analyze something from both directions. So we can look at it from the most primitive cell up to the differentiated cell we want, and we can also analyze from the opposite direction, from the mature cell and maybe back to a more primitive cell. And so that to me is where I see at least the impact that I might have in this field is being able to look at an issue from a different side, because we have this tool, these embryonic stem cells, that we can try to play with to make into the types of tissues we want. Not that those tissues will immediately be therapeutically applicable, but what we learn in this research might provide information to allow future directions to be more likely to result in therapeutic applications. So let's say we learn something really exciting about differentiating embryonic stem cells. Maybe that'll allow us to identify a better population in adult stem cells, or figure out how to take mature adult cells and turn them into what we need them to be. I don't know if that really answers your question, but I have a lot of hope for the area. That's why I'm still here. Yeah, back there.
What's the ability of embryonic stem cells, if you differentiate them down a pathway, like you're saying endothelial or your cardiomyocytes, and you get them there, if you take them back out and put them in a regular growth media, do they
go back? Yeah. We don't have any direct evidence. I think some people have arguably direct evidence. Certainly there's a lot of indirect evidence that might be possible. That might be something we can see in the model. I'm excited to see that, because I would say if we have any indirect evidence, it's that graph of the sort of messy differentiation that goes on, where I didn't include the RT-PCRs, but it's visible in the RT-PCR. The student asked me not to include the RT-PCR. She was embarrassed by them. But it shows the same kind of thing. It's something turns on and then it seems to turn back off again. And it's all relative. So the question is, my theory is, we have this competition thing where the cells that became endothelial cells aren't out competing the undifferentiated cells. It could also theoretically be the capacity of these endothelial cells to go back to some less primitive state and no longer express these markers. We don't know. Or maybe into another cell type that doesn't express these markers. We don't know. And so that's going to be an important question is, what really is going on with these sort of cellular kinetics, things turning on, things turning back off again. But it was a good question.
Following that question, so you choose several kinds of promoter if you rise like day 4 and day 7. So
Right.
in a sense, if you take up the day 4, then compare with the one showing up at day 7, is there any big difference in the population?
We'd like to analyze that. That's why we're trying to go into maybe some more functional assays. From the very crude assays we have, which are things like antibody staining or LDL uptake, there doesn't seem to be a noticeable difference. There doesn't seem to be a noticeable difference in morphology. All of that's extremely crude, though. I think our main question up to this point had been, are they even endothelial? We have now identified that, yes, they're endothelial. Now our question will be, well, how endothelial are they, or what is the difference? What kind of difference does a less mature cell have versus a more mature cell? But we haven't done a lot of characterization of that yet. Be interested in collaborating if you're interested in that area. Yeah.
Yeah. So these markers follow the differentiation. For some cell types, it seems easy and convert earlier, such as 6 days, so we can see the marker. But for some of the other cells, maybe we need to need a very long time
Right.
to differentiate that, and need several different kind of marker.
Right.
So
That's a good point.
Mid-stage, late-stage, different markers.
Right, so this might be a good strategy. He was saying that some markers might turn on in a reasonable period of time, like 3 to 7 days, whereas some markers might take much longer. And this strategy was perfect for endothelial cells because it does turn on really early. I think I probably would have been killed by my student if I picked something that it took 2 months to turn on. But yeah, so I think if we were looking at different tissue, we might have to reevaluate our strategy, or maybe look at more primitive markers. So for example, with the motor neuron project, the promoters we're using are actually extremely primitive promoters, but promoters that are on in these precursors for these motor neuron cells. So maybe the motor neuron cells don't develop until much later, or aren't mature until much later, but we can pick precursors that have a better potential to become motor neuron cells. But yeah, that's an important question, is how broadly applicable would this strategy be? We don't know yet.
With respect to the escrows back in California, I didn't want to let you off the hook on this one, because do you think that they're actually going to be successful in preempting the federal government from actually mandating and having oversight, and that they might actually bite off more than they can chew by doing the chimeras and all the other spots? Are they being successful, or are they?
I have to say I was impressed. So the one of a few take-home messages from that meeting was that California is way ahead of the game, particularly with these escrows. They're way ahead of the game. They have had these escrows up and running for 2 to 3 years easily, and or their scrows, I guess they called them. And will that preempt future intervention? I can't predict that. I doubt it. But it might be the kind of thing where if there's some consistency across the board, that will just be defined as how we need to handle it from now on. So if California, and part of the reason for this workshop was to see cross-institutionally how we can develop some consistency. Maybe California develops some consistency so that the same kinds of things are evaluated and the same questions are answered by their escrows. And then maybe the federal government says, hey, that's the way we all ought to do it. And so the legislation might just be what we've developed over time ourselves. But yeah, what's going to happen in the future? I don't know. We might have a big political shakeup in about a year or so, but we might not. So we'll see.
Any other questions? Thank you again.
Thank you.